This is the myosin thick filament. In this cycle, first of all, the myosin head is locked tightly to an actin monomer in the actin filament. In this stage, the myosin head has a rigor configuration. The configuration is called as rigor configuration because this configuration is responsible for rigor mortis. Rigor mortis is the rigidity of death. In an actively contracting muscle, the locked state of myosin head to an actin monomer in the actin filament is very short-lived. This state is rapidly terminated or stopped by the binding of an ATP. In the next step, an ATP binds to the large cleft on the back of myosin head. This ATP binding immediately causes a conformational change in the domains of myosin head. These domains in the myosin head make up the actin binding site. This conformational change reduces the affinity of myosin head for actin and also allows the myosin head to move along the actin filament. The cleft on the back of the myosin head then closes like a calm shell around the ATP. This closing of the cleft triggers a large shape change. Due to this large shape change, the myosin head displaces along the actin filament by a distance of about 5 nanometer. The bound ATP then undergoes hydrolysis to produce ADP and PI or inorganic phosphate. The ADP and PI remain tightly bound to the myosin head. Due to this ADP and PI formation, the myosin head weakly binds to a new site on the actin filament. This weak binding causes the PI to be released. As a result, the myosin head tightly binds to the new site on the actin filament. The release of PI triggers a force called as power stroke. This force generates a change in shape. During this shape change, the myosin head regains its rigor configuration. During the power stroke, the myosin head loses its bound ADP. As a result, the myosin head returns to a state in which it can start a new cycle. At the end of the cycle, in its rigor configuration, the myosin head again tightly locked to the actin filament. Now, important point. In this cycle, the myosin head remains bound to the actin filament for only about 5% of the entire cycle time. This allows many myosins to work together to move one actin filament. Please like, subscribe and share.